Hello everyone, I am Mahesh and in this presentation we will be discussing about nucleic acid metabolism, and under nucleic acid metabolism we will be discussing about DNA and its replication. The biological information flows from DNA to RNA, which is called as transcription, and it is ultimately the DNA that controls every function of the cell through protein synthesis. The replication process, in which DNA copies itself to produce identical daughter molecules of DNA, which is essential for the survival of the species. Replication is semi-conservative. The parent DNA has two strands complementary to each other. Both the strands undergo simultaneous replication to produce two daughter molecules. Each one of the newly synthesized DNA has one half of the parental DNA, one strand from the original and other half of the new DNA. This type of replication is known as semi-conservative, since one half of the parent DNA is conserved in the daughter DNA. In this video, you will be seeing how the DNA is being replicated.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Origin of replication. The initiation of DNA synthesis occurs at the site called origin of replication. Replication bubbles. The two complementary strands of DNA separate at the site of replication to form a bubble, which is called as replication bubble. DNA replication process is generally referred to as discontinuous because the polymerizing enzyme can add nucleotides only in the 5' 3' direction. Synthesis in one strand, that is leading strand, is continuous in 5' 3' direction towards the fork. In the other strand, that is lagging strand, as the fork opens, multiple sites of initiation are exposed. The synthesis then proceed in short segments in the 5' 3' direction itself. That is, the synthesis in the lagging strand is discontinuous. The short segments of DNA synthesized on the lagging strand are called as Okazaki fragments. These fragments are about 1000 to 2000 nucleotides long. Differentiate the leading strand and lagging strand. Leading strand, it is a replicated strand of DNA which grows continuously without any gap. Whereas lagging strand, it is replicated strand of DNA which is formed in short segments called Okazaki fragments. Its growth is discontinuous. Leading strand does not require DNA ligase, whereas lagging strand requires DNA ligase for joining the Okazaki fragments. The direction of growth of leading strand is 5' to 3'. The direction of growth of lagging strand is 3' to 5' through in each Okazaki fragment it is 5' to 3'. The leading strand only a single RNA primer is required, whereas in lagging strand each Okazaki fragment requires a new RNA primer. The leading strand, the formation of the leading strand is quite rapid, whereas the lagging strand formation is slower. The template open in 3', 5' direction in the leading strand, and formation of leading strand begins immediately at the beginning of the replication. In the lagging strand, the template open in 5', 3' direction, and the formation of lagging strand begins a bit later than that of leading strand. Replication of DNA occurs in 5', 3' directions simultaneously on the both strands of DNA. On one strand, the leading strand, the DNA synthesis is continuous, and on the other strand, the lagging strand, we also call it as discontinuous or retrograde strand, the synthesis of DNA is discontinuous. We'll have to remember that the DNA synthesis is semi-discontinuous and bidirectional. We call it as semi-discontinuous because on the leading strand it is continuous and on the lagging strand it is discontinuous. And we also have to remember that the DNA synthesis is bidirectional. That means on both the sides of the replication fork, the replication takes place. Separation of two strands of parent DNA results in the formation of replication fork. The replication fork moves along the parent DNA as the daughter DNA molecules are synthesized. DNA helicases are the enzymes bind to both the DNA strands at the replication fork. Helicases move along the DNA helicase and separates the strands. The synthesis of a new DNA strand catalyzed by DNA polymerase 3 occurs in 5' 3' direction. This is anti-parallel to the parent template DNA strands. The synthesis of two new DNA strands simultaneously takes place in the opposite direction. One in a direction 5' to 3' towards the replication fork, which is continuous, we call it as the leading strand, and the other in the direction 5' to 3' away from the replication fork, which is discontinuous, we call it as lagging strand. The small fragments of the discontinuously synthesized DNA on the lagging strand are called as Okazaki fragments. These are produced on the lagging strand of the parent DNA. Okazaki pieces are later joined to for a continuous strand of DNA. DNA polymerase 1 and DNA ligase are responsible for this particular process. Polymerase 3 also has a proofreading activity. It checks the incoming nucleotides and allows only the correct matched bases, that is complementary bases, to be added to the growing DNA strand. Further, DNA polymerase edits its mistakes and removes the RNA primer and takes its position. Of new DNA strand is continuous till it is close proximity to RNA primer. DNA polymerase 1 removes the RNA primer and takes its position. The enzyme DNA ligase catalyzes the formation of a phosphodiesterase linkage between the DNA synthesized by the DNA polymerase 3 and the small fragments of DNA produced by DNA polymerase 1. Another enzyme, DNA polymerase 2, participates in the DNA repairing process. As the double helix of DNA separates from one side and the replication proceeds, supercoils are formed at the other side. Over here, in this particular side, you can see like initially there is a uncoiled DNA. Later we have a coiled DNA, which upon further coiling or further formation of the DNA or synthesis of DNA, there is supercoils which is being formed. So this is just like holding a rope on the two hands and continuously twisting it on the opposite sides. If we are twisting it on the opposite sides, initially coiling takes place. Further twisting causes supercoiling. In the similar way, even the DNA supercoiling takes place. The problem of supercoils that comes in the way of DNA replication is solved by the group of enzymes called as DNA topoisomerases. Type 1 DNA topoisomerases cuts the single strand DNA to overcome the problem of supercoils. This we term it as nuclease activity, and then it reseals the strand, which we term it as ligase activity. Type 2 DNA topoisomerases, which is also called as DNA gyrase, cuts both the strands and reseals them to overcome the problem of supercoils. Now, the video demonstrates the complete synthesis of DNA.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
I hope this presentation has made it easy for you to understand the DNA replication. Thank you very much.